So when we think of enzymes, this is what comes to mind. Here's a huge chunk of food. This huge chunk of food gets broken down into smaller pieces with the help of a chemical substance, and that's what we call enzymes. This is what we know from the preliminary level. We also know that enzymes are actually proteins, that is, they are made up of proteins. However, this does not satisfy the answer to the question what enzymes are. What are they actually, and what are they responsible even for? For that, you need to know the definition of enzyme. The definition of enzyme. And enzymes are nothing but just catalyst. And I hope you know what a catalyst is. Let's suppose here's a reactant A which gets converted into product B in certain time duration T. Let's suppose in the same reaction an appropriate catalyst is used. So an appropriate catalyst is used. Upon using this catalyst, the rate of reaction increases, that is, the reaction accelerates, and it takes time less than time t. So catalyst, it increases the rate of reaction. However, the catalyst itself does not take part in reaction. Doesn't take part. So that's what a catalyst is. Since enzymes are catalysts that are secreted by your tissues, which is a biological part of your body, that's why we call this a biocatalyst. So what are enzymes? Enzymes are biocatalysts, and there's nothing else you need to know. Enzymes are biocatalyst. Next, the properties of enzymes. Properties of enzymes. Property number 1. Enzymes are colloidal in nature. Colloidal in nature. Property number 2. Enzymes are thermolabile. Let's say here's your enzyme. You start heating this enzyme. As the temperature increases, the activity of this enzyme decreases, and then there comes a temperature when the enzyme gets completely deactivated. This property of the enzyme is called the thermolability. This is what thermolabile means. Thermolabile means that when you increase the temperature and heat up an enzyme, it becomes deactivated. Number 3. Enzymes are proteins. However, there's an exception to this, and the exception is an enzyme by the name ribozyme. Ribozyme. So this enzyme right here is not a protein, it's an RNA. However, it functions as an enzyme even though it is not made up of protein. Number 4. It is specific. That is, the enzyme acts on specific substrate. So these are the 4 properties. CTPS. Enzymes are colloidal in nature. It is thermolabile, that is, it gets deactivated upon increasing the temperature. They are proteins with the exception of ribozyme, which is actually RNA, and they are specific, that is, they act on specific substrate. Now let's get into the topic that you are actually here for, and that is the classification of enzyme. Enzyme classification. Now, enzyme can be classified into various categories, and they are, let me use a different color, and they are, you have oxidoreductase enzyme, oxidoreductase. You have transferase enzyme, transferase. You have hydrolase enzyme, hydrolase. You have lyase enzyme, isomerase enzyme, and finally ligase enzyme. See, at the end of the name of any enzyme, you'll find a prefix that is ase. Oxidoreductase, transferase, hydrolase, lyase, isomerase, and ligase. And the part before is basically defines the meaning of the enzyme itself. You don't need to memorize anything. For example, let's begin with the first type, that is oxidoreductase. So here's the first type of enzyme that you need to know. Oxidoreductase. So there's is at the end. Before that, oxido and reduct. That means this enzyme is responsible for catalyzing chemical reactions that include oxidation and reduction. That is, the enzymes that are responsible for catalyzing redox reactions are your oxidoreductase. And where can you find redox reaction in your body? Throughout the respiration. The respiration itself is a stepwise oxidation reaction. Throughout glycolysis, link reaction, and Krebs cycle, you can find multiple processes where oxidation is taking place, and each of these processes have their own oxidoreductase enzyme responsible for catalyzing them. For example, let's take link reaction. So in link reaction, as you know, there's your pyruvic acid, and this pyruvic acid gets converted to acetyl coenzyme A. Here, the oxidation of pyruvic acid is taking place in this particular arrow. And oxidation is an exothermic reaction, that is, there is release of energy, and this released energy is stored by NAD in the form of NADH2, and reduction takes place. So in the black arrow, oxidation is taking place. In the red arrow, reduction is taking place. So it's an oxidation reduction reaction. There's an enzyme which catalyzes this reaction, and the name of that enzyme is pyruvic acid. Since dehydrogenation, that is oxidation of pyruvic acid, is taking place, dehydrogenase. This enzyme right here is an example of oxidoreductase. There are various types of oxidoreductase enzymes. So there's your number one, dehydrogenase, which is responsible for dehydrogenation, or shall I say oxidation. Oxidase, responsible for oxidation, peroxidase, and finally reductase, which is responsible for reduction. Okay, so this is the first type of enzyme. Second, number two is transferase. Again, at the end there's is, so it's an enzyme. Since it contains the part transfer, that means this enzyme is responsible for catalyzing the transfer of certain group. So there's transfer of a group. For example, let's take the first reaction of glycolysis. In the first reaction of glycolysis, glucose molecule gets converted to glucose 6-phosphate. Where does this phosphate come from? The phosphate comes from ATP, adenosine triphosphate. From adenosine triphosphate, we get a phosphate. So the triphosphate gets converted to diphosphate due to the decrease in the number of phosphate. And the phosphate that we get from adenosine triphosphate gets added to the last carbon of glucose, and that's how we get glucose 6-phosphate from glucose. Fine. What has occurred here? Here, the transfer of phosphate group has occurred. And which enzyme is responsible for catalyzing this process? It's hexokinase. So hexokinase is responsible for the transfer of phosphate group during the conversion of glucose into glucose 6-phosphate. So hexokinase is a transferase enzyme. There are other transferase enzymes as well, such as first you have hexokinase. Let's not write hexokinase. So of first we have kinase enzyme, which is responsible for the transfer of phosphate group. Up next we have aminotransferase enzyme, aminotransferase, which is responsible for the transfer of, I hope you have already guessed it, amino group. And up third there's your transcarboxylase enzyme, carboxylase enzyme, which is responsible for the transfer of carboxyl group. So that's it for the second type of enzyme, that is transferase. Now let's look at the third type. So the third type of enzyme is hydrolases. Hydrolases. See, at the end of the name there's ase, so it's an enzyme, and it has the part hydro. That means this enzyme is responsible for catalyzing chemical reaction that includes hydrolysis. What's hydrolysis? Hydrolysis refers to lysis or cleavage by the addition of water. So let's say here's a bond between A and B. This bond gets cleaved or broken down by the addition of water. This is what we call hydrolysis, because there is lysis with the help of hydro, water. Example. Can you think of any example? Throughout the process of digestion, there are multiple enzymes that are responsible for the breakdown of large food molecules into smaller food molecules. And these enzymes are responsible for what of food molecules? For hydrolysis of food molecules. So they are what kind of enzymes? They are hydrolases. So starch gets broken down into glucose with the help of enzyme amylase. So amylase is hydrolase because it brings about hydrolysis of starch into, not glucose, not directly glucose, I'm so sorry, into maltose first. Okay, that's hydrolase. Number 4 is lyase. Okay, at the end is, so it's an enzyme. At the beginning, ly, so it brings some kind of lysis. However, we have already learned lysis. We have learned about hydrolysis. See, lyase enzymes are responsible for catalyzing lysis reactions except hydrolysis. So lysis except, I repeat, except hydrolysis. So all those chemical reactions where cleavage is taking place not by the addition of water is, so all those reactions where cleavage is taking place not by the addition of water is catalyzed by lyase enzyme. For example, so here's your fructo 1,6 bisphosphate. This is made up of 6 carbon. Fructose 1,6 bisphosphate gets broken down into 2 3-carbon molecules. One is dihap, that is dihydroxyacetone phosphate, and the other is 3-phosphoglyceraldehyde. This right here is triose sugar, which is ketone because it contains acetone group, and this right here is your aldehyde sugar, which is a triose aldehyde sugar because it contains aldehyde group. What has taken place here? A 6-carbon molecule has split into 2 3-carbon molecule. So we call this step splitting. And the enzyme that is responsible for mediating this splitting or catalyzing this reaction is aldolase enzyme. And this aldolase enzyme is a lyase enzyme because lysis of 6-carbon molecule has taken place. Wait, let me explain you again. So here's a 6-carbon molecule, 1 2 3 4 5 6. Okay, 6. This 6-carbon molecule gets broken down or cleaved into 2 3-carbon molecules, 1 2 3, and another 1, 2, 3. Here, the addition of water does not take place. So it's a lysis reaction which does not include any addition of water. And the enzyme that is responsible for catalyzing this reaction is aldolase enzyme. So aldolase is a lyase enzyme. Fine. Next, number 5. Number 5, isomerase. At the end, there's ase, so it's an enzyme. At the beginning, isomer, that means this enzyme has something to do with isomers. And it actually has. That is, isomerase enzyme is responsible for catalyzing the interconversion of isomers. Interconversion of isomers. See, isomerase enzyme is responsible for catalyzing interconversion of isomers. For example, glucose 6-phosphate and fructose 6-phosphate, there is others isomers. The conversion of glucose 6-phosphate into fructose 6-phosphate is catalyzed by enzyme glucose 6-phosphate isomerase. And this enzyme right here is an isomerase enzyme because it's doing the job of interconversion of isomers or catalyzing the process of interconversion of isomers. Up next is the last category of enzyme, and that is ligase enzyme. This enzyme is also called synthetase. Synthetase. Ligase enzyme is responsible for catalyzing joining of two molecules by expenditure of ATP. Have you seen this somewhere? Anywhere? Maybe in case of DNA. Do you remember it? See, during DNA replication, you have something called Okazaki fragments, right? Here's one fragment, here's another fragment. These two fragments, they get connected to one another. And the enzyme that is responsible for this connection is a ligase enzyme. So what is ligase enzyme responsible for? Ligase enzyme is responsible for catalyzing processes where two molecules are joined together by the expenditure of ATP. And that's it for classification of enzymes. Let me revise the whole thing for you. So here's the enzyme classification. The first type is oxidoreductase enzyme. These enzymes are responsible for catalyzing chemical reactions where oxidation and reduction is taking place. For example, during respiration, there are many steps where dehydrogenase enzymes are responsible for catalyzing redox reactions, and dehydrogenase enzyme is indeed an oxidoreductase enzyme. Second is transferase enzyme, which is responsible for catalyzing the transfer of groups. For example, during the conversion of glucose into glucose 6-phosphate, phosphate group gets transferred from ATP to glucose, and that's how glucose 6-phosphate is made. The enzyme right here is hexokinase enzyme, and kinase enzyme is indeed a transferase enzyme because it is responsible for catalyzing the transfer of phosphate group. Thirdly, we have hydrolase enzyme, which is responsible for catalyzing hydrolysis, that is cleavage of bond by the addition of water. This takes place throughout the process of digestion, where enzymes are responsible for hydrolyzing complex food molecules into smaller monomers. Then there's lyase enzyme, which is responsible for catalyzing lysis reactions other than hydrolysis. For example, aldolase is responsible for splitting of 6-carbon molecule into 2 3-carbon molecules. Here no addition of water is taking place, still lysis is taking place. Isomerase enzyme is responsible for catalyzing the interconversion of isomers. For example, glucose 6-phosphate and fructose 6-phosphate, they are the isomers of one another, and the interconversion of these two molecules is catalyzed by glucose 6-phosphate isomerase enzyme, which is an isomerase enzyme. And finally, there's your ligase enzyme, which is responsible for connecting two molecules together by the use of ATP. For example, during DNA replication, the various pieces of nucleotides, they get connected to one another or joined together with the help of a ligase enzyme. And that's it for the classification of enzymes. Next up, is going to be a video on mechanism of action of enzymes.